Okay, is my screen visible? And am I audible? Okay, thank you for confirming. Okay, welcome. Thank you for joining the second last session for the problem solving session of biochemistry. So today is the second last session and tomorrow will be the last session. And as I have mentioned, today is the last theoretical problem solving session, as in we'll cover topic which is covered in the last week of this course. And we will be going over the two topics or the two broad themes that were discussed in this week. Tomorrow, which will be the last day, which is the last session for this course, what, as I have mentioned previously as well, that what we will be doing is we will be broadly looking into what all we did in this session. It's just a revision session, and I have questions from several assignments of yours and other questions that will basically help you grasp the topic. So I'm trying to reduce the theoretical aspect because we have already covered the theory in the 12 weeks that we have covered. So you would be able to always refer back to those videos. In those videos, we have set up new questions, questions that you have already solved in the assignment. I won't be able to cover everything, but I have picked up some questions that we can discuss, and few of them are numericals as well. So we can discuss that way also, and that will be again at the same time from 6 to 8 tomorrow, right? So do join that session because that will be the last session, and you can also look into the different questions that are important, which are pertinent to that chapter, right? Okay, so with that, let's begin today's session first. Just a second. Yeah. So today's session, we're going to discuss about cholesterol. So last time we discussed about lipid biosynthesis and lipid catabolism. We're going to take up from the same topic, and we're going to continue with the biosynthesis of lipid. I mean, and in lipid, the most important lipid of it is cholesterol. So we're going to look into how cholesterol is biosynthesized, and we're going to see after that I'm going to move into a very interesting topic, which is neuroendocrine system, which is also the hormones which are responsible for most of the cell actions. So we're going to look into where those hormones are synthesized and which are those hormones, what are the categories of those hormones. So those are the broad two topics. And of course, I have questions which are relevant to those topics from previous assignments and previous questions, papers. Okay, with that pre-note, let me begin. So as I remember mentioning last time also that cholesterol is one of the important lipids, right? And we, of course, whenever we say cholesterol, we always think of it in the terms of blood pressure. So whenever we say, like there is high level of cholesterol in the blood, and we say, oh, there are a lot of cardiovascular diseases, which is also called CVD. So you have a lot of cholesterol, so you will have a predisposition towards CVD. But that's the load of cholesterol we know, or we are mostly aware of. What is less discussed is the role of cholesterols in as a component of cellular membrane and as a precursor of steroid hormones and bile acids. So like I mentioned, the second part that we will be looking into, which is hormone, you will see that cholesterol synthesis is very important because it's a precursor for all of those hormones, and we will appreciate that hormones play such an important role in our body. So if we need hormones, we need cholesterol, and that doesn't mean that that cholesterol is always looked into from a very negative perspective. But having said that, cholesterol is, of course, an essential molecule in animals, including us, but it is not required in the mammalian diet. Why I say so? I say so because cells can synthesize cholesterol from simple precursors. Even when we now look into the biosynthesis of cholesterol, one important note that you should remember, cholesterol can be synthesized from very simple precursors, which are already there in our cells from different diet, right? But what is the idea here? That cholesterol is not only contributing to our CVD, it is also a very important component for the cellular structure, right? Right. Now, here on the slide, you can see that something that is structure called isoprene. So this isoprene units are the essential intermediate in the pathway from acetate to cholesterol. So cholesterols are synthesized from acetate, and from the pathway of acetate to cholesterol, isoprene is a very, very important precursor. Okay. But however, just remember that acetate is the beginning point for all the uh, cholesterol that you would see. And if you see here this structure, just a second. Yeah, if you see this structure, it says that acetate is the precursor for cholesterol, and ultimately this one acetate, as it looks like it ultimately, as we will see in the biosynthetic pathway, it undergoes several reactions to form this 27 carbon molecule, which is called cholesterol. And you can see the 27 carbon molecules. You can see 4 fused rings. So important here is that you remember the structure of cholesterol. Okay, so you will see there are 4 fused rings and there are 27 carbon atoms. Okay, and on your right here, which shows you the acetate to mevalonate to squalene and cholesterol, these are the 4 steps how cholesterols are synthesized. It's very simple. We have acetate, as I mentioned, it begins with acetate. Second step is where you have the mevalonate, which gets converted to activated isoprene, which gets converted to squalene. And the squalene, as you see here, it finally undergoes several reactions to become fused in the form of 4 rings. That is the cholesterol structure. Now, with this 4 steps that is there, as we have been doing so far for lipid biosynthesis or the other biosynthesis that we have looked into, we will see how these 4 steps are taking place in a bit more detail. But it, of course, is covered in your course, so you can always look at it into your lecture notes. But let's do it in simple terms, because the idea here is to just look briefly over these 4 steps. But before I do that 4 steps, as I've shown you in the previous, let me go back maybe, previous slide here. So you can see the acetate is converting to mevalonate because of condensation reaction. Then you have the mevalonate, which is converting to isoprene units. Then the third one is basically polymerization reaction of the isoprene units, 6 of those isoprene units, which is forming a 30 carbon linear squalene. So this structure that you see here is a linear structure with 30 carbon. So when it is fusing, it is removing those 3 carbons to become a 27 carbon molecule. So basically in the last step, you have cyclization of squalene to form the 4 rings of steroid nucleus. So this thing here, the 4 fused ring, it's the steroid nucleus. And when you have this steroid nucleus, we will see in hormones, this steroid nucleus has several other derivatives, several other functional groups, which makes a different kind of hormones. But this is the steroid nucleus that we will be talking about. Okay. Right. So coming to the first step now, the first step here is synthesis of mevalonate from acetate or acetate. So in this, as you can see here on your slide, so you have acetyl coenzyme A, which gives you acetoacetyl coenzyme A. This gives you a structure called HMG-CoA, which finally gives you a structure called mevalonate. Now, if you go into the details of this, what's exactly happening is you have 2 molecules of acetyl coenzyme A, which condense. So the first reaction to take place is condensation. So you have 2 acetyl coenzyme A condensing to form something called acetoacetyl coenzyme A, right? Acetoacetyl coenzyme A. Now, this again condenses with the third molecule of 3-acetyl coenzyme A, and that together gives rise to something called HMG-CoA, or if I look into the full form, beta hydroxy beta methyl glutaryl coenzyme A, right? And these 2 reactions are catalyzed by thiolase and HMG-CoA synthase, right? Now, when you will see this name synthase, HMG-CoA synthase, be mindful of 2 things here. One is it has 2 forms, cytosolic and mitochondrial. So both this enzyme is found in mitochondria and cytosol. Now, the cytosolic synthase that we are talking about, it is different from the mitochondrial isozyme. The mitochondrial isozyme that is involved in the synthesis of ketone body. So we haven't done ketone body, but there's also ketone bodies. And for the synthesis of those ketone bodies, the same enzyme is used, which is HMG CoA synthase, but that's in the mitochondria. What we are discussing now, when we are talking about cholesterol, we are talking about cytosol. This distinction should be clear. Okay. All right. So what happened here is, so you have the first step being thiolase, where you have 2-acetyl coenzyme A. This is forming acetoacetyl coenzyme A. Then you have the second enzyme, which is HMG CoA synthase, which is giving rise to HMG CoA. Then, so this synthesis, so we have done the HMG synthesis coenzyme A. Now, what is the third reaction here? Remember, this third reaction here is very, very important. This, if you remember from glycolysis, steps like rate limiting steps, steps like commitment steps. So this is one of those steps here. This reductase step is the uh, rate limiting and the committing step of the cholesterol biosynthesis. What does it mean? That if this reaction takes place, the pathway will lead to the cholesterol biosynthesis, right? So in this step, what happens is the HMG coenzyme A that was formed in the previous step, now this is converted to mevalonic acid. And this is by the use of two molecules of NADPH. And the enzyme is HMG CoA reductase, right? Now, what you need to remember when you are looking into this is, like I will tell you tomorrow also, all these biosynthesis that we have discussed, of course, they are very hefty. They have a lot of pathways, and the ideal way is to remember the pathways, yes, but among them also very important is to remember these rate limiting steps, these commitment steps, because you need to remember these very well. So whenever you're revising or whenever you're looking into the biosynthesis, be mindful of which step is the most important step in that biosynthetic pathway. So for this one, this step here is the rate limiting step. Okay, right. So we have the mevalonate acid from the first step, for the first stage. Now let's go to the second stage, which is basically converting the mevalonic acid to isoprene, but activated isoprene. We'll see what does that mean. Okay, right. So now in the next step, what happens? So in the next step, as you can see, the mevalonate um, has transfer of phosphate group from ATP. Okay, so you have mevalonate. On this, subsequently, you have 3 ATPs giving their phosphate group to ultimately form. Ultimately, when we will see it, the end result, that is the isopentenyl pyrophosphate or the dimethyl allyl pyrophosphate. Okay. So these phosphates that we have just now seen, this attached to the C3 hydroxyl group, as you can see on the figure here. And what happens ultimately is, if you see this group, this structure here, which is called the isopentenyl pyrophosphate, when this is formed, this is one of the 2 activated isoprenes. So this is one of the isoprene which is activated. And this, as you remember in the first slide, we say isoprene is very important for cholesterol formation. So this is one of the important isoprene for cholesterol formation. The second one is basically when this molecule gets isomerized and it forms dimethyl allyl pyrophosphate. That is the second activated isoprene, which is also very important for the production of cholesterol, right? So as you can see here, uh, all these 3 consequence steps which add phosphates here, which is ultimately this is what is forming. Then you lose out carbon dioxide and inorganic phosphate. Ultimately, what you are left with is, you need to remember these 2 names: IPP, isopentenyl pyrophosphate, and dimethyl allyl pyrophosphate. These are the 2 activated isoprene which have these phosphate groups attached on them, which makes them the activated isoprenes. And these come from mevalonate in 3 subsequent steps of ATP or phosphate addition. Right? Now, what is the third step? How do you go from here? Where do you go from here? So now in this third step, what you do is again condensation of these isoprene units to form squalene. Okay. So the isopentenyl pyrophosphate and the dimethyl allyl pyrophosphate as we have just seen, it will now undergo something called head to tail condensation. Here also you have 3 steps. We will see. Here is there are condensation, 3 condensations, but the first 2 are head to tail, and the third one, as we will see, is again you will see that it's a bit different. It is head to head. So there are 2 head to tail condensation, and one is head to head. We'll see all of this. It will make sense in a while, right? Okay. So first you remember that they undergo head to tail condensation, the isopentenyl pyrophosphate and the dimethylallyl pyrophosphate, right? And in this, what happens is, as you can see, you have one pyrophosphate group is displaced, and this forms the 10 carbon chain, which is called geranyl pyrophosphate, right? Now, head to head tail means this and this are not getting attached on each other. They are getting attached at the tail of the other. So head to tail. Okay. So you have your geranyl pyrophosphate. This is forming when one of the phosphate group is leaving, and these are forming a structure with each other. Okay. All right. Now what happens after that? This geranyl pyrophosphate that is formed, this undergoes another head to tail condensation with another isopentenyl pyrophosphate, and now it will. So every time these pyrophosphates are condensing with each other, they are forming each of them a 5 carbon molecule. So when these two are attaching, they're adding 5 more carbons. So when the first 2 happen, isopentenyl and dimethylallyl, 5 plus 5, 10. Then this 10 plus another isopentenyl gives you 15 carbon compound. That 15 carbon compound is called the farnesyl pyrophosphate. Okay. Now, when the farnesyl pyrophosphate has formed, the last step here is two farnesyl pyrophosphate. They will join head to head, and this will eliminate both the pyrophosphate group, and this will form the squalene, as you can see here. So when you have farnesyl phosphate, this is their phosphate, this is the phosphate. Both this phosphate, which is the head group. So both the heads are removed. So both the phosphates are removed, and you have this structure which is connected to each other. This is linear structure, remember. So this is a linear structure which is squalene. So what do you need to remember? Isopentenyl pyrophosphate and dimethylallyl pyrophosphate. Terms to remember are head to tail condensation, head to head condensation, ultimately forming squalene. Okay, and 15 plus 15, farnesyl is 15, another farnesyl is 15, so together they are coming, and that's forming 30 carbon compound. That's what we had discussed last time, right? This is a 30 carbon compound. Okay, now the final step from squalene, how do you go to conversion of this into cholesterol, conversion of this into the steroid nucleus? What happens there, right? So now the squalene molecule, as I told you already, that the linear structure of the squalene molecule forms a cyclic structure to give rise to the sterols, right? So all the sterols, as I mentioned, that steroid nucleus or the 4 fused ring will always be there in any sterols that you pick. Okay, and those sterols have 4 fused rings with a steroid nucleus and an alcohol group at the 3 carbon, and that is by the name sterol, steroid nucleus plus an alcohol group. So stero and all. Anytime you have alcohol, it's all. So sterol. Okay. Now you have your sterol here. So you have the enzyme called squalene monooxygenase. Now this enzyme adds 1 oxygen from the O2 or the oxygen molecule. So 1 oxygen is added to the squalene chain. This addition of oxygen makes it something called epoxide, right? And what happens is this epoxide, which is also called the squalene 2,3 epoxide, it undergoes several reactions. Concerted reaction means several reactions together. So several reactions that can cyclize the entire structure, and this gives rise to the steroid nucleus that we are talking about. Okay. This cyclization, now you will see here that there are many fates to this. So this squalene 2,3 epoxide, it forms in plant, it will form stigmasterol. In fungi, it forms ergosterol, and in animal, it forms the cholesterol. So as I mentioned, from simple precursor, you can have cholesterol. Now, cholesterol is mostly found in animals. in plants and fungi, they have their kind of cholesterol. So this name you should remember. In plants, you have stigmasterol, and in fungi, you have ergosterol, right? So after this squalene through 3, so as I mentioned, these fused ring is important to the sterol or steroids. So this will be there. The only difference is if you see in this and in the final structure, this has a C2H5, and this has some very minor bond changes. There's a double bond here. There's no double bond here. So this is why ergosterol, this is stigmasterol, and in animals you find cholesterol, right? Okay. Uh, yeah. So that's how your cholesterol's biosynthesis takes place, right? Now, what happens to these cholesterols? You have made its body has synthesized is fantastic, but what is it doing? What is the purpose of it? What is the entire? As I have already mentioned, it has several roles that is being what it's being employed or used for. So, as we know, mostly much of the cholesterol synthesis as it takes place in liver, and from there, a small fraction of that is incorporated in the membrane of these liver cell, and most of it, however, is exported outside. So few of them go to the membrane, which is come part of the lipid biomembrane. But I mean, some of these are carried, and most of these are carried out, and they are formed or they are exported as different other components. So what are the different components of cholesterol that you see? You see cholesterol as bile acids or their salts. So these are hydrophilic high cholesterols. So they are synthesized in liver, of course. And bile acids, if you remember, they are also part of digestion of lipids. So they are exported out for digestion purpose. Second, they can be exported as cholesterol esters. So they can form esters in the liver, and so basically they are this enzyme is called acyl coenzyme A cholesterol acyl synthesis. You don't have to remember, but what I'm trying to say is that there is a transfer of the fatty acid from coenzyme A to the cholesterol, forming a cholesterol ester. But what is the importance is these are transported and secreted lipoprotein to other tissue that use cholesterol. So the cholesterol is transported in this form so that the other cells and tissues can make use of it, right? So it can be either given out as bile acids, or it can be exported as esters to carry to the target tissue where they can be utilized, and they're carried as lipoproteins. This is a few technicalities that you need to remember, right? This is the ester that you see forming here, right? Now on your right, this table here shows you the different kind of lipids that you have. So you can have chylomicron that we have already discussed. You can have VLDL, which is very low density lipoprotein, or you can have low density lipoprotein, or you can have high density lipoprotein. And all these, sorry, uh, yeah, and all these are different types of, you know, uh, lipoproteins or different type of usage of cholesterol. And when there's a question that specifically deals with these kinds of classes, so there I will tell you how are each of these differing from each other. So for time being, just remember that they are classified as these different kinds of lipoproteins. Okay. Now this is something that is part of regulation of cholesterol biosynthesis, and not very difficult concept to understand, but you need to be mindful of what is the cascade that is taking place here, right? Now, so as we have so far been discussing that uh, there is always a feedback inhibition, there is always a regulation by the end products, that's always there, we always know. And one of the crucial step that is always regulated in any biosynthetic pathway is the rate limiting step, and that is exactly why you should know the rate limiting step, because that helps you understand which of the enzyme in that pathway is the most important for regulation. If you regulate that pathway, if you regulate in that pathway, if you are able to modify that enzyme, the entire pathway gets affected. So that's why you should know the rate limiting step and the enzyme and the substrate in those directions. Right? Right. Now, So let's talk about our biosynthesis of cholesterol. And we know the most important rate limiting step here was the conversion of HMG CoA to mevalonate with the reaction catalyzed by HMG CoA reductase. In that step also we had several enzymes, but the step that was rate limiting was the third step, which was by HMG CoA reductase. Right? So regulation in response to cholesterol levels is mediated by the transcriptional regulation of this gene, HMGA CoA reductase. Because if you don't transcribe the gene, then you don't have the protein. If you don't have the enzyme, then your entire pathway is regulated. That is what is the idea here, right? So what happens here is there is this very elegant pathway or elegant mechanism called SREPB activation, and we'll see what happens here. So this gene, HMG CoA reductase, is basically controlled by this small protein family called, so I'll tell you the full form once only so that we don't have to get confused over it again and again. So the name SREPB is from sterol regulatory element binding protein. If we break the name, it says sterol regulatory element binding protein. So there is an element in the DNA sequence which is regulating the control, regulating the synthesis of sterol. So sterol regulating element, and the proteins that bind to this are called the binding proteins. So sterol regulatory element binding proteins. So these are what is the SREPBP that is we're referring to. Now I will read it out once. I will make you like go through the figure once, and then I will once again come and describe this a bit slowly. Okay. So now, when newly synthesized, these proteins that you are seeing here. Yeah, sorry. Yeah, right. So when these proteins are newly synthesized, they are embedded in the endoplasmic reticulum or the ER that's shown here, right? So they're embedded in this ER. Now, only the soluble amino terminal of the domain of these proteins, they are the transcriptional activator. Like I mentioned, they are regulatory element binding protein. So any protein that binds to a regulatory element is basically, try to understand it this way. This regulatory element, and this is where the gene transcription point, which is the transcription starts, right? So this is the transcription frame, and this is where the regulatory element is. So if any protein binds to this regulatory element, it's basically a transcriptional activator. It could be a transcriptional repressor also, but in this case, it's a transcriptional activator. So this protein binds to this element here. Element is basically DNA sequence. It binds to DNA sequence, and it enhances the transcription of the downstream gene, which is the gene for HMG CoA reductase, which is an important enzyme or important pathway step for cholesterol biosynthesis, right? Right. So when it is synthesized, it's only embedded in the ER here. It's here, right? It's here. Now, only the amino terminal, which is again a soluble form of this protein, only that can go and bind to the transcriptional activator. Makes sense. Why? Of course, because if it's embedded in the membrane, how will it move inside and go to the DNA? So it has to be soluble so that it goes inside the DNA. So while it is newly synthesized, that is not happening. Right? Now, this domain, however, has no access to the nucleus, and this cannot participate in gene activation while it remains as part of the SREBP molecule. So the SRBP molecule is here. It's in the ER. While it is bound here, it cannot go and bind to the DNA, right? So something has to happen such that it detaches itself, and that element or the protein that is able to bind to the DNA sequence moves inside the or is able to access to the DNA. So how is that happening, right? So to activate this or to make sure that this interaction is taking place between the protein and the DNA element, this domain, which is transcriptionally active domain, it separates from this SREBP complex by proteolytic cleavage, right? So there is a cleavage happening here, which breaks this up. And as you can see here, this breaks happens, and this soluble domain, which is there, this moves inside, moves to the nucleus, binds. So this break. Now, who is breaking this and why is this breaking? How is it breaking? This is what is important, and this is why this slide I kept so that I can explain this in detail. Right? Okay. So as we have understood the concept here, that it is when it is bound to the ER, it cannot go and bind to the nucleus. It has to break away, break free from this. Now, when cholesterol levels are high, you already have a lot of cholesterol in the cell, then your these SREBPs, which are on the ER, they are inactive, right? They're secure to the endoplasmic reticulum, so they will not break away, but they are bound to another protein called SCAP, which is SREBP, cleavage activating protein, right? Now, so where is this protein located? Where do you see in this red color guy here is a protein that binds to these protein, but it has a domain which is the cleavage activating domain for this protein. It will, because I'm reading for the first time, it might sound a bit difficult. Let me just complete it and then we'll come back and it makes more easier. If you have any doubt at that time, you can let me know. Right? So at this junction, when it is synthesized and when it is on the ER, it is bound to this cap also. Right? Now, this cap, however, is a sterol sensor. Right? Why? Because it can bind to cholesterols. Right? Now, when sterol levels are high, this complex of SCAP and SRDPB, they are interacting with another protein, maybe, that retains the entire complex in the ER. Okay. Now, let's do the opposite. When the level of sterol in the cell decline, which is when you don't have cholesterol and now you need cholesterol, some conformational change happens here in the SCAP and the SRDPB, and the SCAP as a complex detaches from the ER and goes to the Golgi complex, as you can see on the slide. Now, in the Golgi complex, this SRAPB is supposed to be cleaved, and the cleavage happens there. So there it is cleaved in two steps by two proteases, right? The second cleavage releases the domain which is important, and that domain transfers or travels to the nucleus and binds to the DNA and does the enhancement of the transcription. So the whole idea here is, as you can see, the level of cholesterol themselves regulate if the synthesis needs to be taken place. Let me put it in this very simple way. If cholesterol levels are too high, you don't need cholesterol in the cell. There will be no synthesis or no gene transcription. And gene transcription, you don't want gene transcription, so you would like this entire complex to be retained on the endoplasmic reticulum. Because if that doesn't happen, then if it binds and it moves and it goes to the Golgi complex, it will break away and move and synthesize more cholesterol, right? But when your cholesterol is low and you want cholesterol, this here breaks away, moves to Golgi complex, is cleaved by two proteases and it moves inside. Now, how do you sense? How does this sensing of cholesterol take place? It takes place by the same protein. It is a sterol sensor. It can sense. It basically is able to bind. If there is cholesterol, it is bound, so it stays. If there is no cholesterol, it doesn't bind, it goes away. It is as simple as that. But it is important for you to understand that how is this minute changes taking place, so that when you are looking into the figures and when you're revising, it doesn't confuse you. It's not confusing. It might look like a very complicated figure, but it's not. It's just a movement of a complex from endoplasmic reticulum to Golgi complex for further downstream activation of the gene. Right? So this was just one of the steps that is regulated. And as so far we have already looked into this, that there are several steps in the biosynthetic pathway that is under regulation or under the regulatory check. So here also, we remember, right, we started from acetyl coenzyme A, we went to HMG-CoA, from there we went to mevalonate, from there we went to cholesterol, and from there, as I have told you, that we have LDL, VLDL, all these different kind of cholesterols. So all the crosses here, as we have discussed, all the crosses basically stop this pathway. So if you have glucagon, this basically stops the synthesis of cholesterol. If you have more of these cholesterols, of course, then it will inhibit the pathway. If you have cholesterol formed as cholesterol esters, so if there is more cholesterol, it will favor the formation of esters, of course, because it wants to move. And if it is insulin, it would favor this entire pathway by enhancing or favoring this reaction of conversion of mevalonate. So basically, the idea is the same wherever you look into regulation, and this is what we have been telling so far, that if you know the pathway, you know the checkpoints of your regulation. But where do you start? You start with the rate limiting steps and the most important committed steps of that pathway, right? This is not a very difficult pathway to describe, so it was pretty simple. Now, from the cholesterol, as I told you, the steroid nucleus is the basis of the hormones that we will see. So cholesterols form something called pregnenolone. From there you have progesterone, and from there you have cortisol, corticosterone, testosterone, which are again further several other hormones. And all of these hormones, as we know, have several functions, and we will do that in the next half of today's session. So you can see how important cholesterol is. As we discussed, it can move as bile acids, but it can also move to other tissue for the synthesis, for the production of these hormones. So they are very important when they are produced, right? And these steroid hormones, again, like just an example of how the pregnenolone is formed. So the cholesterol is formed, if this ring you see, on this structure, you have modification, and that gives rise to the pregnenolone, which again further undergoes several modification to give rise to different kind of hormones, right? And this is exactly what we are trying to show here with this image here. Cholesterols can give rise to bile acid. We have discovered this. We know that steroid hormones is what is also derived from cholesterol. And at the same time, you have several vitamins. If you see here, D, E, A, K, all of these are connected to cholesterol. So cholesterols come from IPP. IPP also give rise to these different kind of compounds. Cholesterol give rise to these compounds. We should appreciate the importance of cholesterol and not just think from the perspective of CVDs or cardiovascular disease, is what this image is trying to say, right? And in about hormones, we'll do it in details in a while. But before that, let's try to grasp this topic a bit more. So let's try to solve these questions. And yeah, similar. So, one note before I quickly go to these questions. A lot of assignments of ours in this course were all about correct or incorrect kind of questions, which basically tells you that you need to be very well versed with the topics. Today also you have a lot of questions like this. Tomorrow's session, which will be the concluding session, where I will be trying to cover different questions. I would like to focus more on numericals, but at the same time, there might be some questions which are correct and incorrect based questions. So I will try to keep numerical so that that gives you a hands-on experience. Because the theoretical part, which is correct, incorrect, or this kind of questions, I'm pretty sure when you are revising, you would be able to come up with the answers. Numericals is what, or numericals, or problems where are many, there are examples and names, is the kind of question that I will be focusing on tomorrow, which will be the last session. All right. Yeah. So coming to today's question answers, which will also, I think, cover mostly theoretical only and match the following. Yeah. So let's come to the first one, which is which of the following are precursor or intermediate in cholesterol synthesis? A, B, D. Perfect. Because we have already discussed all these steps here. We have looked into mevalonate, we know farnesyl pyrophosphate, we know squalene, we know all of these are basically the precursor of one another, right? And we have not encountered pyruvate even once in this pathway. We have encountered acetyl coenzyme A, but not pyruvate, right? Okay. Next, in an experimental knockout mice lacking SCAP, which of the following will occur? So SCAP, remember what SCAP does, and if you remove SCAP, what can happen? So where is SCAP and what is SCAP doing? And if you remove SCAP, what can happen? That's it? Anyone else? Or any other option other than A? Okay. The answer is A, of course, and C. So A is, so basically, if you remember SCAP is bound to SRD PBP. SCAP is also bound to cholesterol. If it doesn't bind to cholesterol, then it moves away from the ER to the Golgi, and then it breaks it away from the SRD-PB, and that moves. So basically, if there is SCAP, it is not binding, right? If SCAP is there, it is binding to SRBP and sensing the cholesterol level, right? Now, if you are removing SCAP, then of course that movement of the complex from the or the protein from the ER to the Golgi complex doesn't take place, right? So that's why the first one is there, because it is not moving to Golgi anymore, it will remain trapped in the ER. And the third one says LDL receptor expression will be reduced. LDL is basically low density lipoprotein. Now, because you have less of these synthesis, and there is less of cholesterol, and there will be less of LDLs, there will be less of LDL receptors too, because because there is no cholesterols that would require uptake. So these kindly lipoproteins are important for uptakes of the fatty acid, sorry, the cholesterols. And since you have less cholesterol, there is no uptake. You don't need this. Their activity or their transcription will also be reduced. Right. So these are the correct answers. Now the second one says cholesterol biosynthetic enzyme will be upregulated. No chance, because SCAP doesn't directly like directly control the biosynthetic pathway. The path, the enzymes, the biosynthetic enzymes are upregulated maybe because of cholesterol decretion, but it's not related to SCAP. And there is no excess cholesterol because you don't have the cholesterol synthesis, right? Okay, next one. Okay, which of these compound inhibit cholesterol biosynthesis?
Statins.
Okay, that's statins. Anyone else? Any else option?
Oxysterols.
Okay. Okay, so that's A and D, and someone says A and B. So any other answers, or A, B, and A, D? I'll take that. Okay, so let me give the answers. A, B is also correct, as well as D is also correct. Fantastic. As you have rightly guessed, statins is inhibiting the reductase, so HMG-CoA reductase, and as we know, that is very important. So it inhibits cholesterol biosynthesis. Glucagon, we have already seen in the pathway in the regulation network that glucagon negatively controls the biosynthetic pathway. Oxysterol, it inhibits the SREPB activation, so it's part of the SRBB and SCAP complex. While insulin, we have seen in the same regulatory figure that it actually activates the biosynthesis pathway, right? Okay, next one. A patient that has a mutation that inactivates the reductase enzyme. So which are the likely these are the likely outcomes.
ABC.
Okay. That's it. Just a second. Anyone else wants to answer? I have one answer, ABC with me. Anyone else? I'll wait for the other person to also try to attempt and then we can discuss. Okay, ABC. All right, wait a second. ABC, as you would have already understood why, because if there is no reductase, there is no synthesis of cholesterol. So you don't have mevalonate, you don't have steroid hormones, and you have acetyl coenzyme A accumulation because that's not being used up and that's not being getting converted into its further subsequent product. So that makes sense. And if this is correct, then this is opposite of it. It doesn't make sense at all. Right? Good job. Okay. Which of the following would reduce intracellular cholesterol levels in the hepatocyte? So basically, in liver cells, which of these will reduce the cholesterol level?
D.
Okay. I have D. Okay. I'll wait for the other person as well. In the meanwhile, you can check if you have any other options or it's just the only answer. Anything else? Okay, B. Okay, so the answer is statin administration, yes, and PCSK9 overexpression. Now, this enzyme, I will just get the full form so that I don't get it wrong. It's basically called, it's a proprotein convertase subtilisin caxin type 9. So this enzyme is important because it degrades the low density lipoprotein receptors. So this overexpression is basically it's reducing your receptors synthesis. So because it's overexpressing, there will be more degradation of the receptor. Because there is more degradation of the receptors, there will be less cholesterol uptake in the liver cells. So there is no cholesterol level increasing. So it will reduce the overall concentration, concentration. And statin administration, as we have already discussed, statin is inhibitor, right? C is basically true because so if you have loss of LDL receptor function, uh, it's basically reducing the intracellular level because again you cannot take it inside, you cannot uptake the cholesterol. And insulin, as we know, it promotes cholesterol synthesis, right? So it cannot go with this in hand in hand. Right. I'm sorry, this one. Now the next one is choose the incorrect statements regarding cholesterol. This again. So this is the kind of question that I was saying. This is very prevalent in the assignments where you will have a lot of these incorrect and correct questions, which is basically part of your. So do attempt these kind of questions because these kind of questions help you. There is one thing, it's more prevalent in the course and it is also helping you grasp the topics.
Option E.
Mm, low density lipoproteins are rich in cholesterol. Okay. So that's incorrect, is what you're saying. So the question says incorrect. So when you're answering, be mindful if you're looking in the correct statements or the incorrect statements. The answers here. Okay, I'm wait.
And the D also, madam.
Okay. Um, anyone else is answering? Okay, 10 more seconds and then I will discuss the answers. Yeah. A and D. Okay. Well, correct. D was correct and A is also, so basically D is incorrect and A is incorrect. Rest of the statements are correct is what the answer, the meaning here is. So let me look into this. The first one says, owing to the requirement of cholesterol in membrane, dietary supplementation is necessary in mammals. This is the first thing that we discussed. When I started today's session, I said that cholesterol is important, yes, but it's not required in the diet. It's not a thing that you should have in the diet. It's not essential because cholesterol can be synthesized from very, very simple precursor like acetyl coenzyme, which you already have or which is an intermediate of several other pathways. So it's not a supplement or it's not an essential component in our diet. So even if there is requirement in the membrane, it doesn't mean that it has to be supplemented by the dietary component, right? That's why it's incorrect. Second one says all carbons of the cholesterol molecule come from isoprene precursor, which is correct. We have already seen that. C says cholesterol derived bile acid can be conjugated with glycine or taurine, which is also correct because this increases their solubility and in the emulsification of the fat. So it's basically helping them in solubilization, right? Fourth one says here that triacyl glycerols require chylomicrons for their transport owing to their high hydrophobicity, whereas cholesterol molecules are freely transported because of their hydrophilic side chain, which is incorrect. Because cholesterol also is not freely transported. This is what I was telling. So for transporting the cholesterol, you require lipoproteins. Remember the lipoproteins that I said I will discuss later. So the lipoproteins, which are chylomicrons or LDL or HDL, as so far we already we have, I think we have understood that this is important for cholesterol uptake. So your LDL, HDL, chylomicrons, these are different kind of lipoproteins which enable the movement of cholesterol, which enable the transport of cholesterol, right? Cholesterol, because it is hydrophobic, even though this has this hydrophilic side chain, overall it is hydrophobic. So this is not transported freely. Not cholesterol, not even cholesterol esters, that is the form that it is converted to. Neither of these forms are transported freely in the blood. Like for DHs, you need kilomicrons. For cholesterols, also you need kilomicrons, LDLs, and HDLs. These are the proteins that I said that I will discuss later in the question. This is one of the questions here on previous questions we've already discussed. They're important for the carrying of the cholesterols. And the sixth one says the very same thing, that the LDLs, which is low density lipoproteins, they are rich in cholesterol and cholesterol ester, because that's is what their primary constituent is. They are carriers of cholesterol in the blood, right? Very cool. Okay, next one. Mitochondrial HMGA. So this is the distinction between mitochondrial and the cytosolic component that we had discussed in the first few slides, right? Yeah, so I'll wait for you people to come to the answers and then we will discuss. This one should be simple. This is not that difficult. Okay. Others? Yeah, that's just one option. Any other option? So B, any other option? E, C, D, F. Okay, let me give the answers. Sorry. A, B, E, and F. Yes. So I think I got, what did I get? No, I got C, D, F, which is okay. Okay. And I also got B and E. All right. So I got a mixed answers. You can check in the slide and let me tell you which ones are not correct or which ones are correct. The first one says mitochondrial HMG-CoA synthase is involved in the synthesis of ketone bodies. Correct? Because we discussed that the mitochondrial ones are in the ketone bodies or for the ketone bodies, and the cytosolic ones are for the cholesterol biosynthesis. Right? Right? Good. Next one says mevalonate synthesis is the committed step in cholesterol synthesis. This we have already established. The step is by HMG-CoA reductase. Right? The enzyme here is reductase, and that's why D is incorrect. D says HMG-CoA synthase catalyzes the rate limiting. No. Synthesis is the previous step. The rate limiting step is the third step at the first stage, which is by HMG-CoA reductase. So that's why it is incorrect. D. C says statin inhibits HMG-CoA synthase. Basically the same thing. It is not synthase, it is reductase, the rate limiting step. So be mindful of the synthase and reductase. That's the only thing I think where we have made this mistake. Right. Next one says two head to tail condensation reaction takes place for the formation of solid. Yes, because even though there are three condensation steps, two of them are head to tail. The third one is head to head, right? Farnesyl and farnesyl pyrophosphate, right? Right. And the last one says plants do not synthesize cholesterol. That is correct, because as we saw, cholesterol is in animals. Plants produce stigmasterol, and the fungi, they produce ergosterol, right? Good. Okay. Next one. So we have cholesterol transport and different activities of cholesterol. So looking at this option, which ones do you think are incorrect? So which of these options, if you read and if you compare, it is incorrect. Be mindful, it is incorrect. Yes, see, what else? F, C, F, okay. CNH, FCFCH. Okay, that's it. Let me give you the answers and then you can check. So the incorrect statements of these 8 are A, B, D, and H. H, I think I got the answers. Let me see the others, or let me show you why the others are incorrect. But the first one should have been very, like very easy to have come across, right? So cholesterols increase membrane fluidity. That's an incorrect statement. So basically, cholesterols have two role. That's why I wanted this. This is important. When you go at higher temperature, they decrease the membrane fluidity by restraining the phospholipid movement. So when you have higher temperature, it decreases it. When at lower temperature, that time it prevents solidification. That time it increases membrane fluidity. So just saying it increases membrane fluidity, it's basically an incorrect statement. It has to be specified with the temperature as well. Is it at low temperature? Is it at a higher temperature? If it is a higher temperature, then it decreases the fluidity. If it is lower temperature, yes, it increases the fluidity. Okay, that distinction has to be made. Next one says, compared to high density lipoproteins, chylomicrons contain more phospholipid and cholesterol esters, which is incorrect, because chylomicrons, the other category of carriers, they are rich in triacylglycerol. So they carry the TAG. This we have already discussed, right? The HDLs, which is the high density lipoprotein, they have phospholipid and cholesterol esters, which are more in than TAGs. So that's why it's incorrect. So chylomicrons carry the TAG, high density carry the phospholipids and cholesterol esters, and the LDLs, as we have discussed, they are also rich in cholesterols and cholesterol esters, right? The LDLs, the HDLs, in addition, contain the phospholipids as well, right? The C, third one, is incorrect. It's correct. It says its chylomicrons are synthesized in the ER, which is endoplasmic reticulum, and they are transported by a lymphatic vessel. That's a correct statement. Okay. Fourth one says a loss of function mutation, which is basically, so such a function means if this mutation takes place, the activity of the protein is lost. Right. So a loss of function mutation in ApoC2 will promote premature TAG release. So ApoC2 is basically activator of lipoprotein lipase, right? And this lipoprotein lipase enzyme, this basically hydrolyzes TAG, right? So a loss of function mutation in this ApoC2 leads to impaired TAG breakdown. So that's why you have these TAG breakdown, which is taking place, another release, right? So there's this release and find a difference between breakdown and release. The actual term is breakdown of the TH, right? Fifth one says a similar function in ApoE. This inhibits endocytosis of chylomicrons by hepatocytes, right? So if you look into your notes, you will see that these proteins and these terms are given, ApoC2 and ApoE. Okay, all right. Next one says release of free fatty acids from TAG of the VLDL increases the density of lipoprotein complex, which is correct, because as your VLDL lose TAG, they become more small. They become smaller. They become denser. So they convert to something called low density lipoprotein, from very low density to low density lipoprotein. So uh, they basically increase the basically they become smaller in size and they become denser in the uh, mass. Okay. Seventh one says lipoprotein endocytosis require the binding of LDL receptor to ApoB. So again, when you look into the names, you will or notes, you will see that all these proteins are mentioned there. So ApoB 100 is basically required for the binding of LDL receptors, right? And the last one says endocytosed LDL receptor undergo lysosomal mediated degradation. This is incorrect because when you see your endocytosis pathway, you will see that the LDL receptor, they are actually recycled back to the cell surface. When the LDL, so basically when the LDL delivers the cholesterol, or whenever it delivers the substrate to the lysosome, these most of the LDL receptors are cycled back to the surface and not may undergo degradation. So they are not degraded. So basically, this is something referring to how you are cycling the receptors. Sometimes the receptors are degraded as well with the uh, ligand, which is also targeted for degradation. Most of the time, the receptors are recycled back in this case, right? That's the distinction. Okay, next one. Now you have cholesterol metabolism. Which of these statements are correct?
Except C and F, all are correct.
C and F. Okay. Did I get any other option? Okay, I will wait for a few more seconds for the other person to answer. So you're saying except C and F. Okay. Okay. A. Okay. All right. So you can type your answers while I am also showing the answers on the slide. Right. So the correct answers are A, B, E, and F. Okay. And we'll see why F as well. So first one is correct. I think everyone already mentioned that when you have positive regulation of cholesterol synthesis, it of course depends on the transcription regulation that we have seen because of the SREBP proteins, right? Second one says a gain of function mutation in the sterol binding domain of the SREBP cleavage, which is cap protein. This inhibits cholesterol biosynthetic genes. Yes, because if it is binding to cholesterol, the idea is if you are binding to cholesterol, then you are retaining more of these SREBP complex in the ER. It doesn't go to Golgi complex. It doesn't break. The transcription domain doesn't go inside the cell to nucleus. Right? So that's correct statement. Next one states phosphorylation of the rate limiting enzyme of the cholesterol biosynthesis uh, activates is this is incorrect because phosphorylation of that gene actually it inhibits it, so it inactivates it. Phosphorylation stops the reaction. Fourth one says high level of cellular cholesterol positively regulate LDL receptor gene transcription, which is incorrect. So if you have high cholesterol, you inhibit the LDL receptor gene. This is by the expression of SREBP activity, suppressing the activity. So you basically are reducing the cholesterol uptake if you have high level of cellular cholesterol, right? Because that's how it's retaining. E says high level of cholesterol promote cholesterol acidification. Yes, because if you have cholesterol, you want to transport it, you make it more acidic. Sixth one says high blood glucose promote cholesterol because high glucose means high insulin, and we have seen that insulin positively controls or positively regulates the cholesterol biosynthetic pathway. So more glucose, more insulin. That's why it is correct, because glucagon is the opposite. Glucagon is basically doing the opposite of insulin. So you have more insulin when there is more glucose, and insulin positively regulates the glucose, the cholesterol biosynthetic pathway. Okay, the last question of this segment. So you have to choose the correct statements for cholesterol derived hormones.
A, B, C, I mean A, B, D, E are correct.
A, B.
D, E.
So C is not correct. Okay. Anyone else? A and E. Okay, let me give you the answers. Sorry. A is incorrect. Otherwise, everything else is correct. Okay. The first one is progesterone is a delta 5 derivative of pregnenolone. It is incorrect because when we will see, it is basically a delta 4 derivative of pregnenolone. Okay. The otherwise statements are all correct. Loss of function mutation in the 3 beta hydroxy steroid dehydrogenase. It will inhibit the formation of all steroid. Basically, if you see in the, wait a minute. Yeah. If you see here, you will see that when we look into the enzymes, this enzyme is important enzyme for the conversion of these different or the formation of these different hormones. So that's why, sorry, if this enzyme has a loss of function, it will inhibit all the formation of the steroid hormones, right? C one says the production of glucocorticoids occur in mitochondria, which is true. Fourth one says estrogens are 18 carbon derivatives of androgens, which is also correct. And the last one says patients with loss of function of 17 beta hydroxy steroid dehydrogenase will have low testosterone because this is the enzyme that converts, or this is forming the testosterone. So if you have a loss of function, basically the function is reducing, so you have low testosterone levels. See, these names are almost similar, but they have the different compound that has been formed at the end. If it becomes 17, it is specifically for testosterone. If it's 3 beta hydroxysteroid, it is for all the steroid hormones, right? Okay. Okay, now let's quickly go to our next topic, which is hormone. And I will not take much time because even though it's very interesting, it's mostly you need to know the names of the hormones. You need to know the like where they are synthesized, how they are synthesized, how in a sense under what control. So those I am not going to cover in much detail, but what I'm going to cover is overview of how these hormones function and what is the role of these different kind of hormones. Right. All right. So we'll begin by neuroendocrine system, which we are all very much aware of. It's basically the junction where neurons and the endocrine cells or endocrine system come together for the formation and transport of these hormones, which are very important, which have specialized functions in different tissues and different organs, right? So these hormone signals, they integrate, they coordinate the metabolic activities of different tissues, and they not only are, as we will see, are important for several functions like for your food or for your digestion. It has very vast roles, the hormones, right? Now, in comparison or in relation to this, there was this topic this which was discussed in the notes, which was called radio immunoassay or the RIA. Now, the RIA was developed by, or it received the Nobel Prize. Uh, this was this is a technique which is basically extensively used for hormone research, right? And as you would have already seen, the principle is somewhat similar, or the principle of ELISA is derived from this technique, right? So the RIA, why is that? Because radio immunoassay uses radioactive compounds, which are, of course, which are not safe for usage. So a newer variation of RIA is the ELISA that we use. So if you know ELISA, if you have read about ELISA, these concepts are very, very similar. If you haven't, we will just go over it quickly, right? So the idea here is to know how much of your hormone is there, how much of the hormone has been secreted, how much of the hormone is synthesized, right? And for that, you use something called antibodies, which are specific to the hormones. So what you do is you have a purified hormone which is injected into rabbits, and the rabbits, the hormones are basically the foreign particles for them, so they elicit antibody towards that hormone, and these antibodies bind to that hormone very specifically. So when you uh, collect, or when you isolate this antibody from rabbit, you have these antibodies which are very specific for a particular hormones, right? So you have these antibodies. Now what you do is you have a constant amount of this antibody. You know how much antibodies you have collected or how much antibodies you are using, right? So when you use this constant amount of antibody and you incubate it with a fixed amount of radioactively labeled hormone, right? So now you know, let's say this much unit of receptor is there and this much unit of hormones are there, and they will bind with each other. Yes, and they are radioactively labeled. So whenever this binding is happening, I should be able to detect it, right? So that incubation you have done, right? And you have a certain fraction of these active hormones, radioactive hormones, binding to the antibody. Now, what is the usage here? If with this radioactive or radio labeled hormone, you are putting labeled, unlabeled hormone, right? So the unlabeled hormones will compete and will displace some of the labeled hormones from its binding site of the antibody, because there will be a competitive binding between the labeled and the unlabeled, and the labeled will displace the unlabeled will displace the labeled, right? So this binding competition is what is quantified with a reference of standard curve. And from that, you get to know how much of the unlabeled hormone is there in the solution, which is basically the degree of the labeled hormone displacement uh, from the antibody, which gives you a measure of how many unlabeled hormone is there. It is RIR is radioactive because it is using the radio labeled hormones, which are the ones here red. The blue ones are unlabeled, whichever have been displaced by the how do you say, the unlabeled, the blue ones. They are binding to the blue and the reds have been displaced. So you basically, after this binding, you do the you check the radio active levels, and you get to know how much of the radioactive level the hormones has been removed, and that with that, using a standard curve, we get to know how much of these unknown sample had how much of the hormones. So this technique is important because it also is what is used in ELISA. It basically uses the principle of competitive exclusion to understand how much the concentration of one particular entity is there, right? Okay. Now, basically, hormones, when we talk about hormones, there are two kind of hormones. You have peptide hormones, which are predominantly peptidal in nature or proteinaceous in nature, and you have steroid hormones. So when you have the peptide hormone or the amine hormone, they bind to the receptor outside the cell, and they act through the receptor without entering the cell. That's a basic difference. Steroids, because they are steroids, they are entering the cell membrane. Peptide do not do that. For peptide, you have the receptor on the cell surface, which they bind. Steroids enter the cell. So how does peptide hormone work? They will bind to the receptor, and then there will be several downstream components, downstream proteins that will ultimately carry the how do you say, carry the signal to the desired target. Whereas in steroids or thyroid hormone, they will enter the cell and they will directly form the hormone receptor complex wherever they want. And with the ultimate goal is to go to the nucleus and activate the genes, right? So the ultimate goals or the consequences of such kind of receptor interaction of hormone receptor. So this one we have established, right? If steroid hormone goes inside, it will bind to its specific gene. But what happens when you have the hormone receptor interaction? You can have change in the membrane potential because of binding of the hormones to the receptor. So your hormone has bound to some receptors and you have these change in potential. Your, let's say, your sodium ions are moving out and calcium ions are moving inside, or potassium ions are moving inside. So you have an opening of the gate and you have these changes in the ions across the transmembrane layer, right? So that can also happen. Or your receptor enzyme is activated by the extracellular hormone. So let's say your receptor is an enzyme in itself, and the binding of the hormone activates it, and then it has several how do I say, catalyzing activity that it will be doing after binding to the hormone. Or you have second messenger generated inside the cell, which acts as an allosteric regulator for one or more enzymes. So basically, here, if you see in the previous slide, sorry, you have a secondary messenger, let's say CAMP. So this CAMP basically could bind to several other enzymes, regulate this, or enhance the activity or decrease the activity of several other components, right? Or the receptor, which if does not have enzyme, then it will recruit other protein kinases or other proteins. So if it not itself an enzyme, it will recruit other proteins or other cascading signal cascading effects, right? It can also be an adhesion receptor on the cell surface, and this can then interact with molecules in the ECM. And not why does it always have to be inside the cell? It can also be outside the cell. Can be the cytoskeleton as well. And if a steroid molecule causes a change in the level of one or more genes, that's mediated by nuclear hormone receptor. Again, like I said, like we have seen in this figure also, here also the binding is via the receptor and the hormones. So this receptor has to be there. Receptor is basically what binds to the hormones, right? Okay. Now, this is what I wanted to say, that you have these plasma receptors, you have nuclear receptor or steroid receptor, which move inside the cell, right? And they are doing transcriptional regulation. they are producing secondary messages. The type of the hormones can be peptide, catecholamine, icosanoid, steroid, vitamin D, and you should remember one or two of examples of each of these types and where are they synthesized from, right? If you open any hormone figure or any neuroendocrine system figure that we have the image coming up, you will see that the same principle lies everywhere. Which gland is releasing which hormone, which is affecting which, and that hormone is impacting which hormone by affecting the gland. Basically, we'll look at the axis also. We'll see in a while. Right. So if you, so what I would suggest you is, for most of these types, you should remember the precursor, right? For example, catecholamine, which are the epinephrines and the norepinephrines, they are derived from the tyrosine. So this you should remember, right? Then you have nitric oxide, which is derived from arginine, and the enzyme here is nitrogen oxide synthase. Then you have eicosanoids. These eicosanoids, there are three types: prostaglandins, thromboxane, and leukotrienes. You should remember they are from the arachidonates or arachidonic acid. Next, you have steroid. We have established this already. This is from cholesterol via the first intermediate, which is progesterone, which also has pregnenolone. Okay. Then vitamin D, which is by these component 7-dehydrocholesterol, right? And then you have retinoid, which is from carotene. So what I'm suggesting you is you should remember these precursors here, right? This is what I was telling you. You have these endocrine glands, hypothalamus, pituitary, thyroid, parathyroid, adipose, adrenals, pancreas, kidneys, testes, and ovaries, right? So these are the endocrine glands. These are the glands that synthesize hormone, and they will release them into a gland or into a duct, uh, sorry, into blood, and they are then carried to the target cells. You also have something called paracrine hormones. So these paracrine hormones, they are basically released into the extracellular space by the cells, and then they move. So this is not released into the blood. And then you have something called autocrine hormones as well, which are basically targeting the same cell that it is being generated from. So the word autocrine. So autocrine, paracrine, exocrine, endocrine, you should remember. So these are the types that you should remember. And similarly, there's what something called exocrines. So you should also look up for exocrine glands, which are basically they will secrete their substances into a duct. So that's duct release, but that's not what we are concerned with. We are only concerned with endocrine glands, which are basically glands that release their hormone in the blood, right? And then you have very important axis, which is hypothalamus, which is basically passing on the information to the pituitary gland here. The pituitary gland here has two components, anterior and posterior. It's very easy to remember what the either of is doing, uh, because posterior is only responsible for the release of vasopressin and oxytocin. Whereas every hormone releasing factors come from the anterior. So what is releasing is basically anterior pituitary is not itself synthesizing. It will release something called releasing factors, which will go to thyroid. So thyroid releasing factor will go to thyroid, and from there the hormone will be synthesized. So you will have most of these as releasing factors. We'll see it in this here itself. And the next one, we will see how does that axis look like. So this is what I was saying. That let's say you have some response, some trigger, and your central nervous system goes to the hypothalamus. Hypothalamus secretes releasing factors, which goes to the anterior pituitary, which synthesizes something called tropins, all these different tropins, and they go to their targets and the desired target from which the hormones are secreted. And from there, it goes to the target tissue. Let's consider one axis. So from hypothalamus, you have releasing factor, let's say called anterior pituitary. Uh, the releasing factor go to anterior pituitary. From there, you have corticotropin. Corticotropin basically is adrenal cortex stimulating hormone. So you have these hormone going to adrenal cortex, and from there you have the secretion of cortisol or the corticosteroids and aldosterone, all these different. Similarly, if there is thyrotropin, tropin is from the anterior, it goes to thyroid, and all the different hormones are released. So this is all anterior pituitary, while you have these posterior. So via neuronal connections, the signals move from hypothalamus to posterior pituitary, and you see they're directly releasing oxytocin and vasopressin, so they don't have these other targets. So posterior pituitary is only involved in the release of vasopressin and oxytocin, and the rest everything is under the control of anterior pituitary, right? And this is the basically how it is regulated and how it is working. So basically, in endocrine system you have hormonal cascades, as we have just seen. At every step that we have just moved through, right, you will see that they are all enhancing the signal. So be it your corticotropin, be it at this step, be it at this step, all these steps are enhancing the signal. That's because they're targeting different kind of substrates, or they're reaching different kind of targets. So your signal level is increased, and that's how the endocrine system function. And because at every step it is increasing, every step is a possible feedback inhibition, and every step can regulate the overall pathway, like you can see here. So you have hypothalamus goes to corticotropin, which goes to anterior pituitary, where you have adrenocorticotropic hormone, which goes to adrenal gland, and all these. Now, at every step, if you have cortisol, it will stop both the points of secretions. But if you have less of these, it will stimulate the central nervous system, it will stimulate the glands. So because every step is cascade, like I said, right, there's a rate limiting step that can be regulated in hormones. Because every step in the pathway is enhancing the signal, it's basically all these steps in the pathway are uh, they are up for regulation. They can all do feedback inhibition, and they can all control the levels of hormones, right? Okay, we will quickly do the questions. They are all very easy, shouldn't be very difficult. So let's begin. uh, yeah, what is the answer for this? Okay, it's basically talking about amino acid derivative are basically peptide hormones, epinephrine and thyroxine. Which of these are true for these hormones? Okay, maybe we'll quickly go to the next questions. The first one, so the answer here is tyrosine derivative. They all act, they both can act by nuclear receptors. Basically, they can move inside, or they can or they can be via membrane receptors, and they are stored in vesicles. Right? What is incorrect is they are lipid soluble. They are not lipid soluble. They are all you know uh, soluble. They are all protein in nature, so they are all soluble. Right? Next one. What happens if the feedback inhibition of cortisol on the hypothalamus is lost? So I told you, right? So the hypothalamus, just think in the opposite way. If there is cortisol, it is now giving feedback inhibition to hypothalamus. What all will happen? A, B, and D. Fantastic. Yes, that's the exactly answer. So if your feedback inhibition by cortisol is not there anymore, then you will have keep having increased CRH, increased ACTH. Every gland will keep producing more of the releasing hormones and the tropins, and you will have hyperplasia because, of course, there will be more of accumulation. But what you won't have is decreased adrenal cortisol production because the cortisol production will keep on increasing. There is no inhibition of cortisol production, so your cortisol production will keep on going on. That's correct. Very well done. Okay, a very interesting question. You have several type of hormones on your left side and the cells and the hormone names on your right, and there are several master pairs, right? So which ones of these are correctly paired? Take your time, look into the answer. It's not very difficult. The rest of them are very easy to do. Okay, so yeah, you take your time. I'm just reading out the options. So on your left, we are discussing about the type of hormones. We have tryptophan derived hormones, peptide hormone, hormone which have 20 carbon molecule. Try to remember this. There is negative regulation by glucagon. You have endocrine hormone, tyrosine derived hormone. We know these compounds, and we have amine hormone. And on your right, we have different kind of cells, different kind of hormones. For example, serotonin. We have acinar cells. We have nuclear receptors. We have T3, T4. So all of these are already matched. You need to tell me which one is what. Yes, definitely. I will definitely help you. Okay, let me give you the answers and then I'll move you through one of them. So, except for the fourth and the sixth, all of these are correct, right? So, the first ones, let's look at these option wise. Let's look at A first. A says A is to 3. So, tryptophan derived hormone is to serotonin, which is correct because tryptophan derived hormone is serotonin. Okay. So, if you go back on these slides here, so here you will find that there are several other examples which I haven't discussed. So this question is a way of you to look into the names of different kind of hormones that are there. So tryptophan derived hormone is serotonin. So A3 is correct. C is to 6. C is a hormone derived from a 20 carbon molecule. The name of this hormone is leukotriene And this says sixth, which is leukotriene. So that's correct. Okay. E is endocrine hormone, which is so many, which says 9. Norepinephrine. We already have seen this, right? We know it's correct. G is amine hormone, which is basically it's saying it as the melatonin, which is also correct because melatonin is an amine hormone which is derived from tryptophan. So it is correct. So all the options here are correct. So the question says you need to choose options which all correctly paired matched pairs, which is correct. Okay. Next one. Next one says B, which is peptide hormone 211, which is parathyroid hormone, which is correct. Parathyroid hormone is a peptide hormone. Okay. D, which is your negative regulation of glucagon, happens by the delta cells, which is also correct. Okay, because these cells release something called somatostatin, and this somatostatin does a negative regulation of glucagon. Okay, which inhibit the glucagon. So this is correct. Then you have E, which is endocrine hormone, which is again parathyroid. We have already looked into these options here. And F, which is tyrosine derived hormone, which is 9th, norepinephrine, that is also correct. That's a tyrosine derived hormone. So if you see here, we've already discussed. They were catecholamines, they were derived from tyrosine. So this option is also correct. So B is also correct. All of them are correctly paired. Option 3, B, peptide hormone to calcitonin. Correct, absolutely correct. C, which is again hormone derived from 20 carbon molecule. You have the 13 prostaglandin. If you remember arachidonic acid, let me take you back so that you get a flow here. Arachidonic acid, prostaglandins, and leucotriene. So the 20 carbon molecule is arachidonate. That's also correct. E here says endocrine hormone to from the delta cell, which is also correct. We have just established this. And G, which is amine hormone, which is your tree, which is serotonin, which is also correct. Okay. So this is also not correct option. Let's go to the next correct option first. So the fifth one says A, again tryptophan derived hormone to 12, which is melatonin, which is correct. B, which is your peptide hormone to 10, calcitonin, which is also correct. E, which is your endocrine hormone to 8, which is T3 and T4. We have seen this is a thyroxine, which are correct. And G is 9, so amine hormone to norepinephrine is also correct. Okay, so these are also all correct pairs. Now we will look into the question which option which do not have correctly paired, and we'll see what happened there. So the D1, C, which is hormone derived from 20 carbon molecules, and says 11 incorrect. We have just seen the 3 molecules which are derived from arachidonic acids are leukotrienes, prostaglandins, and the this thing. Sorry. So leukotrienes, prostaglandin, and as we have just seen here, thromboxanes, right? So when you see this here, 11 is not a part of that. Now E1, E says endocrine hormone, 1 is acinar cells. So acinar cells are actually exocrine, they are not endocrine. So they don't secrete their into the hormone into the blood or their products into the blood, right? So this is an exocrine gland. This is the type of an exocrine gland. F, which is tyrosine derived hormone, and it says that it is 12, which is melatonin. Melatonin is a tryptophan derived hormone, right? That's not correct. And G is 8, G is amine hormone, which is going to T3 and T4. We know it's not correct because we have just seen which ones it's correct too. Okay. Okay. That's why all of these are incorrect actually here. Sorry. So G, so I'm so sorry, I just realized. So your G, which says amine hormone and it says T3, T4, it is actually correct. But because not all of them are correctly paired, these are, the option is incorrect. Now the last one says again C, which is 20 carbon molecule to 7 calcium regulation, incorrect because those three are not part of calcium regulation, right? E, again endocrine, which is 2 calcitonin, not correct. I'm sorry, which is correct. F, which is tyrosine derived hormone to melatonin, which is tryptophan derived, which we also have discussed previously. And G, amine hormone to melatonin, which is is also correct. But because not all of the options are correctly matched, this is not correct answer. Was this helpful? Too much of explanation, but did you get it? This is a very good question for you to remember which hormone is from which cells and what are the rules. Yeah. Like I said, recording is already always available, so you can always come back, stop, and do take this when you are trying to revise or look into the options. Okay. Next one. Quickly. A person has been fasting for 36 hours. What would you likely see in that person? Which hormonal pattern? A and B. Uh, Yes, A and B definitely because that's what we already know. C also because there is an increased cortisol also due to the fasting. Uh, GH actually is also increased because if GH increases, lipolysis increases, and you in fasting you want more of your fuels to be used up, so you have more of GH increased secretions, and cortisol because it's a response of the body thinking that there is no food, so I need more of these different components. Right. Quickly. Now, a tumor causes overproduction of somatostatin. So, if remember what is somatostatin inhibiting and what is the downstream effect? Okay, so I will give the answer so that we move to the next answer quickly. This is easy. So somatostatin, the speciality or the importance of this hormone is that it is an universal inhibitor. So anytime you say somatostatin, just remember that everything is going to be decreased because it's an universal inhibitor. So in this option also, everything is decreased when there is somatostatin overproduction. So if insulin decretion, TSH decretion, GH decretion, everything is decreased because it inhibits all of these components, right? Okay, next. This is important and this is what is also relevant. So if what is the feature that differentiates peptide hormones from steroid hormones? Let's do this quickly. So remember when we discussed about which receptors these particular hormones bind to, that will give you a lot of cues here. A, okay. Any other option? Okay. Well, A is correct for sure. We also have B and C correct. So peptide hormones are stored in vesicles and steroids are synthesized on demand. That's correct. Second says steroids have longer half-life in circulation. Yes, and that's how it can enter the cells. And the fourth one is also correct. Peptide use secondary messengers more frequently. Remember we discussed, right? Because it's on the cell surface to transfer the signal, it will use different secondary messengers. Third one is incorrect because it says it bind to nuclear receptors. That's incorrect because of the second point. Steroids have longer half-life, so it can enter enter the cell and reach the nucleus. Peptide hormones, however, have very short half-life. That's why they quickly act, need to act quickly, so they bind to the membrane-bound receptor. Okay. All right. Next one. So this is based on RIA, radio immunoassay. This will help you understand this topic better. Okay. Uh, this is the correct answer for this. Well, so let's look into these options. The first one says radioimmunoassay can be used for relative only, but that's incorrect because you can also use it for absolute quantification. Second one is also incorrect. It says that RIA cannot be used for steroid hormones. That's incorrect. It is actually used mostly for steroid hormones. C says chromatography and electrophoresis both can be used for purifying hormones. We have discussed this, right? They can be used for purifying as well. Fourth one says is administration of crude pancreatic extract will not reduce glucose levels in diabetic animals, which is incorrect because it has been shown that if you use crude pancreatic extract, they do lower blood glucose level because they contain insulin. Fourth, last one says oral administration of TRH to TRH deficient animals will potently induce the release of TSH. This is incorrect because it cannot be orally administered. TRH. TRH is actually a peptide, in fact, a tripeptide, and they are not stable in the GI tract, so you cannot orally administer them. Okay. Okay, so I have three questions remaining, which are not very difficult. We'll quickly do this, and we'll spend just one minute on each of these questions. Right? Okay. This is the next question. I would like you to look into the question and answer this based on what you understand. So you have to tell me which of these statements are true. Okay, so A is the only incorrect. Rest everything is correct. I'll tell you why. The statements are we are to look into true statements. So the second message mediated amplification occurs only at the receptor step. That's incorrect, right? Because when you look into the cascade of hormone, you will see secondary messenger targeting another messenger, then it goes to other messenger. So there is many downstream target for the secondary messenger. It does not only happen at the receptor step. So that's incorrect. And other than that, all the statements are correct. So your hormones do mediate cytoskeletal changes. We have discussed this. When your receptor proteins bind to the hormone, they also act outside the cell on the ECM proteins, right? You have steroid molecules that can act directly as transcription factor. That also we have seen already, and we have already discussed this. Steroid molecules can also act via plasma membrane receptors. That's also there. You can also have steroid molecules which is binding to the receptors on the plasma membrane. And lastly, protein or peptide hormones are fast acting because they are short lived, so they directly act with the receptor, while steroids are far more stable, so they move inside the cell and reach the nucleus. So that can take a longer time, right? Great. Next one. Again, quickly, which one of these is incorrect with relation to signal, hormone signaling? Okay, few more seconds and then we look into the answer. Yes, sure. So in this, the incorrect statements are these. So the first statement says anterior pituitary hormones can inhibit glandular secretion. So they also can release inhibitory hormones. Okay. Second one says negative regulation of pancreatic secretion by blood glucose is mediated by hypophysis. So this is incorrect because the blood glucose levels, they directly regulate pancreatic hormone by, like for example, insulin or glucagon secretion is the direct regulation of blood glucose level. They do not involve pituitary glands. So hypophysis is basically pituitary glands. Okay. Next, concentration of respective hormones increases when moving from the hypothalamus to the target gland in the signaling pathway. Correct, because we have just the last thing that we discussed was cascading effect. For every step, your hormone is increasing. Basically, the concentration and the signal is increasing, right? Fourth one is all released hormone regulate their secretion by acting at the glandular, hypophyseal, and hypothalamic levels, which is basically what this means is the hormones are acting at all the three levels, which is incorrect. So feedback inhibition does take place at multiple level, but hormones generally act locally without, like targeting every other pathway. So if the hormone is from this particular like let's say for the cortex, then it will only regulate cortex. This will not go higher up. So it is not that it regulates at the glandular, then the hypophyseal, which is basically the pituitary, and then hypothalamic, it doesn't go there, right? So it acts locally, is what is the statement. Why is it incorrect here? And the last one is severing the hypothalamus hypophyseal portal system will inhibit antidiuretic hormone mediated water retention, which is vasopressin. And vasopressin uh, is via the posterior pituitary, and that does not come under the portal system. So vasopressin, as we know, does not come under the portal pituitary, portal system, and that's why if you are disrupting the hypophyseal portal system, then the vasopressin does not get affected, and this is incorrect, right? Okay, and quickly the last statement for today, last question. This is all these questions. When you revise, it will be easy for you to like grasp the topics quickly. So you're basically supposed to look into the statements, and it's basically related to the synthesis and the transport, and give the truer statements. Okay, since we are less on time, I'll quickly give the answers. You can always check if you were thinking in the correct direction. So the first one says the source tissue of peptide producing hormone has fenestrated capillaries. So when you look into it, you will see that endocrine glands that produce a peptide hormone, for example, pancreas or anterior pituitary, they have these fenestrated capillaries to allow rapid hormone release into the fenestrated is basically window-like system, right? Okay. Then neural tissue produces adrenaline and noradrenaline. It's correct because it's from the adrenal medulla. It is secreted from the adrenal medulla. Third one says eicosanoids are synthesized and stored as membrane compounds. That's not correct. They are, because they're steroid hormones, they're secreted and they're synthesized only on demand, right? So they cannot be, they're not stored as per se, right? Okay, the next one says hormones which are responsible for parturition are synthesized from membrane phospholipids. That's correct. Those are for prostaglandins, if you know that. The prostaglandins, which are important for labor and parturition, they are synthesized, as we know, from arachidonic acid, which are membrane phospholipids, right? Sixth one is also correct. It says neurohypophysis synthesizes vasopressin, which is basically your posterior pituitary, and we saw how they have neural connections. Sixth one says loss of function mutation in cytochrome P450 gene will impede steroid hormone biosynthesis. That is also correct because most of these cytochrome genes, they are involved in the conversion of the cholesterol to steroid hormone. So they cannot, if the cholesterol, the cytochrome gene is not functioning, this will not happen. And the last one says cholecalciferol is an active form of vitamin D, which is incorrect. It's actually the inactive. So cholecalciferol is inactive, and for getting activated, it has to become calcitriol. So it is not the active form of this vitamin D, right? Okay, right. So that brings us to the end of today's session. And if you want to ask any question, you can ask, or we see each other in the next session, which is tomorrow, and we'll discuss the entire course once again. Very briefly, we'll focus mostly on the questions. Yeah. Thank you so much. Thank you. See you tomorrow.